Topics in post-harvest technology. Respiration. Most commodities are harvested at peak quality. Quality can only go downhill. Post-harvest technology, therefore, involves techniques to maximize the shelf life of commodities while maintaining their quality and ensuring food safety. A majority of post-harvest techniques involve reducing a commodity's respiration rate. Respiration has been defined as all those metabolic processes, the sum of which is a net gain of energy to the cell. It involves degradative, also known as catabolic, cellular processes. Many plants store excess energy as starch, which is a polymer of glucose molecules. When needed, the starch is depolymerized into glucose, and through respiration, the glucose is broken down to release CO2, plus ATP, a high energy containing molecule, plus water and waste heat. ATP is also known as adenosine triphosphate, and it is the main energy molecule for animals and plants. Though excess energy is often stored as starch, in some plants the excess energy is stored as fat. Fat is very energy rich, but much more difficult to break down than starch. Respiration represents a loss in dry matter, or, if you prefer, a loss in food reserves. However, respiration rate is not related to the amount of food reserves in plant tissues. Asparagus, which has a very high respiration rate, has relatively low reserves of starch in its tissues. In contrast, sweet potatoes, which are very high in starch reserves, have a very low respiration rate. So, what factors affect respiration rate? First, the age of the tissue. Is it a young, rapidly growing tissue or a tissue that is mature? The type of tissue also has a great effect on respiration rate. This table shows the relative respiration rates of various plant tissues. Notice at the top the general pattern of respiration rate. Young leaves and flowers have a greater respiration rate than fruits and stems, which have a greater respiration rate than storage organs. Flower buds such as broccoli and rapidly growing shoots such as asparagus have high respiration rates. Developing seeds and immature fruits can also have high respiration rates. Unripe fruits like cucumber have a medium respiration rate. The respiration rate is lower for these fruits because they are picked when relatively grown, but before they have begun to ripen. Mature fruits like pumpkin and the winter squashes, think of butternut squash and acorn squash, have low respiration rates. These fruits naturally last for long periods in storage. Storage organs like potato and onion and garlic also have low respiration rates. The respiration rate of a leafy vegetable varies depending on the growth stage when harvested. Spinach, for instance, is often picked as a rapidly expanding leaf and has a high respiration rate. In contrast, a cabbage head, which is a storage structure, has leaves which are more mature, and the respiration rate of this structure is usually low. Temperature greatly affects a plant's respiration rate. The warmer the temperature, the faster the respiration rate, and the increase in respiration is exponential. Very low levels of oxygen will reduce respiration rate. Above normal levels of CO2 will also reduce respiration rate. However, above 2 to 3% CO2 for many plants, CO2 becomes a metabolic poison. So, if you do want to store with elevated CO2 to help retard respiration, you have to monitor the CO2 level to make sure it doesn't get too high. Wounding causes ethylene production, which promotes respiration. So wounding will elevate respiration while the tissues are healing. Plant tissues that are affected by plant disease will have elevated respiration rates. Plus, the plant disease organism itself can have a high respiration rate. Though more subtle in their effects, cultivar, production area, growing conditions, and growing season can all affect respiration rate. Remember, cultivar is a contraction of cultivated variety. We tend to use variety when we mean cultivar, but variety is actually a botanical term that has a precise meaning. So in science, we prefer to say cultivar. With respect to growing conditions, are they good for growing? Is the plant subjected to some stress, whether it's water, nutrient, or temperature stress? These can affect respiration rate. Growing season refers to winter-grown versus summer-grown crops. What are the primary respiratory pathways? There are three we're going to talk about. Glycolysis, the Kreb cycle, also known as the TCA or tricarboxylic acid cycle, and the electron transport chain. Glycolysis is a sequence of enzymes located in the cytoplasm of cells. When starch is depolymerized to produce glucose, the glucose is acted upon by the glycolytic enzymes to yield pyruvate molecules. Here we have a schematic showing the glycolytic pathway. We'll start with glucose in the upper left. It undergoes a number of enzymatic changes, finally resulting in 2 pyruvate molecules. So we go from a 6-carbon glucose molecule to 2 3-carbon pyruvate molecules. The end product of glycolysis may change depending on the oxygen concentration in plant tissues. With relatively high oxygen, from approximately 1% to around 21%, pyruvate is produced when glucose is metabolized. However, if the oxygen level is less than around 1% in the plant tissues, ethanol, also known as ethyl alcohol, will be produced instead of pyruvate. This process is referred to as fermentation. With fermentation in plants, 1 glucose molecule is converted into 2 ethanol molecules, plus 2 CO2 molecules, plus 2 ATP molecules, and waste heat. Fermentation represents a stress situation for plants. Lots of glucose is being shunted through glycolysis to produce enough ATP to keep the plant cells alive, and is energy wasteful as the energy potential of the glucose molecule is underutilized. Also, if the ethanol accumulates to too high levels in the cells, it can actually poison and kill them. The accelerated glycolysis that occurs during fermentation results in abundant CO2 production. Remember, we're channeling glucose very quickly through glycolysis, so it's producing a lot of CO2. Think about wine production. The grape juice is being fermented and lots of CO2 is being produced, which must be allowed to escape. True fermentation occurs under very low oxygen conditions, usually when oxygen is less than 1% in the tissues. Like glycolysis, the Krebs cycle is an enzymatic sequence. However, it is a membrane-bound system located in the mitochondria of cells, unlike glycolysis, which occurs in the cytoplasm. Pyruvate from glycolysis is converted to acetyl-CoA, which then feeds into the cycle. Here is a schematic for the Krebs or tricarboxylic acid cycle. In the upper left-hand corner, notice that pyruvate is converted into acetyl-CoA, and then acetyl-CoA is combined with oxaloacetate to form citrate. A number of enzymatic changes then occur that involve the formation of different organic acids. The cycle restarts when citrate is once again formed. Notice also that we're getting NADH being produced at 4 points in the Krebs cycle. NADH is a molecule that is high energy. This NADH is fed into the electron transport chain to produce ATP. Krebs is a primary source of reductants for the electron transport chain. The electron transport chain, or the ETC, is a membrane-bound enzymatic system also found in the mitochondria of cells. In this figure, we see the mitochondrion. Notice that the Krebs cycle is positioned to feed reductants directly to the electron transport chain. There are a number of protein complexes that are associated with the electron transport chain. Electrons are shuttled from complex to complex, and in the process, ATP is produced. We refer to an electron cascade in the ETC because the electrons, as they're being transferred, are losing energy. That energy is causing the formation of ATP. This is a figurative representation of what's happening with the ETC. As the electrons move from complex to complex, there is a loss of energy, represented by the downward steps, which is being used to produce high-energy ATP molecules. Note that the final acceptor of the electrons is oxygen. The oxygen then combines with hydrogen ions to produce water. So this is why most plants and animals need oxygen to survive. The oxygen is not burning the glucose, which is what we tend to think of as happening in our bodies. Instead, at the end of the ETC, the oxygen accepts the electrons. Without sufficient oxygen, both the ETC and Krebs cycle stop functioning. Essentially, the systems back up. And rather than getting lots of ATP being produced, ATP production goes down. What does the plant do? It feeds lots of glucose through glycolysis to produce enough ATP to keep its cells alive. Under ambient conditions, which are 20.9% oxygen and 0.41% CO2, one glucose molecule yields 2 ATP from glycolysis and 34 ATP from the Krebs cycle working with the electron transport chain. How do we measure respiration rate as CO2 production? For many years, the best method was to use gas chromatography. This is a lab method and not usually used in the field. What the industry does use, especially in storage facilities, is infrared light. Carbon has a valence of 4, so for carbon dioxide, it forms 2 bonds with each oxygen atom. These double bonds very readily absorb infrared light of approximately 4 micron wavelength. With an infrared analyzer, a sample of air is passed through an infrared light source. The double bonds absorb the light and the absorption is measured. The more CO2 that is present, the more infrared light that is absorbed. These instruments are very accurate and can easily measure CO2 at the parts per million level. We can also measure oxygen consumption as an indicator of respiration rate. This is also done using gas chromatography. Other methods are to use paramagnetic analyzers, polarographic analyzers, and electrochemical analyzers. However, as a rule, you will find that most people measure respiration as CO2 production rather than oxygen consumption because it's easier to do. How do we express respiration rate? Well, we could do it as mils or milligrams of oxygen consumed per kilogram of product per hour, or mils or milligrams of CO2 produced per kilogram of product per hour. Again, mils or milligrams of CO2 per kilogram hour is most often found in the literature. Summary points. Most post-harvest techniques involve preserving the initial quality of a harvested commodity. Quality maintenance is most often accomplished by reducing a commodity's respiration rate. Respiration can be affected by many factors, but the primary factor is temperature. There are three main processes involved in energy production in plants: glycolysis, Krebs, and the ETC. Fermentation often represents a stress situation and is most often caused by low oxygen levels in plant tissues. Respiration is most often measured as CO2 production over time and is often written as milligrams of CO2 per kilogram hour.